Okay, so now we're going to talk about capillaries and tissue perfusion for the third recording of blood vessels. Capillaries are generally found in clusters referred to as capillary beds. They wind their way between the cells of most tissues in the body. Blood flow to tissue through a capillary bed is known as tissue perfusion. This is tightly regulated to ensure that the metabolic needs of all tissues are met at all times. Capillaries are extremely thin vessels, with walls that are only about 0.2 microns thick. Each capillary consists of only endothelium rolled into a tube and a small amount of basal lamina secreted by the endothelial cells. Structural and functional features will include periocytes. These are cells found in some capillaries that have contractile filaments and appear to control blood flow through the capillary. And on average, capillaries have about a 15 micron length and about an 8 to 10 micron diameter. The walls of most capillaries will consist of 1 to 3 endothelial cells joined by tight junctions. They will also curl around the capillary's entire circumference. And here you can see a general structure of the capillary. Nutrients, gases, ions, and waste must be able to cross the wall and travel between the blood in the capillary and the tissue cells. The movement of materials is capillary exchange. Materials are exchanged via 3 mechanisms, including 2 types of diffusion and transcytosis. Diffusion and osmosis through the gaps and pores of capillaries. Some capillaries will have small pores within their endothelial cells. These are referred to as fenestrations. Water, as well as small substances, such as monosaccharides, amino acids. These are things that can dissolve in water and are able to move freely through these pores when there is a gradient that exists. And this is how nutrients and things move, is they will move down concentration gradients. Lipid-soluble substances such as oxygen, carbon dioxide, and certain lipids can generally enter and exit the capillary by diffusing across the membrane of one side of the endothelial cell and out the membrane of the other side. Substances that enter either the interstitial fluid or the blood. Transcytosis, this is larger molecules must cross through the endothelial cells by transcytosis. Molecules are taken into the cell by endocytosis, and then they are moved out through the other side of the cell by exocytosis, and this is what the process of transcytosis is. There are 3 types of capillaries: continuous, fenestrated, and sinusoidal. And here's a sort of breakdown of the 3 of them. Continuous capillaries have tight junctions between the cells. Okay, they are the most restrictive as far as what can pass through, or another way to say that, they are the least leaky. Fenestrated capillaries, you will see these fenestrations, and these would be found in the kidneys, endocrine glands, and small intestines, and they are moderately leaky. And then you have sinusoidal capillaries, which are found in the liver, lymphoid organs, bone marrow, and spleen. These are the leakiest. They will also allow large substances such as cells to cross the capillary walls. So the majority of capillaries in the body are referred to as continuous capillaries. They are located in muscles, skin, and most nervous and connective tissues. Continuous capillaries, as I mentioned, are the least leaky, and they permit the fewest substances to enter or exit the blood by paracellular routes because of their endothelial cells, which are joined together by tight junctions. Fenestrated capillaries, these are much leakier than continuous capillaries because their endothelial cells contain fenestrations or pores. The pores allow diffusion to take place much more quickly than it does in continuous capillaries. Fenestrated capillaries are therefore located in places that require substances to rapidly enter or exit the blood. The sinusoidal capillaries, these are the leakiest capillaries, the name often simply shortened to sinusoids, and they have discontinuous sheet of endothelium, irregular basal lamina, and a very large pores in their endothelial cells. These are typically 3 to 4 times larger in diameter than other capillaries, often have irregular shape because of their boundaries and are determined by the organ in which they reside. Sinusoidal capillaries are located in organs and tissues such as the liver, spleen, lymphoid organs, and bone marrow, which facilitate the transfer of large substances, such as blood cells and large proteins, between the interstitial fluid and the blood. The flow of blood takes place within the body's capillary beds is collectively referred to as microcirculation. This involves two types of vessels: true capillaries, where the materials are exchanged, and small central vessel. True capillaries form interweaving networks with multiple anastomoses, fed by a proximal end of the central vessel, which is formed by either small terminal arteriole or metarteriole. Each capillary metarteriole junction contains a precapillary sphincter. This precapillary sphincter controls the amount of blood flowing into the capillaries. And at rest, only about 20% of the body's capillary beds are actually fully open. So here you can see this would be a metarteriole. This is an arteriole. This would be a metarteriole here. And you can see this is the central vessel going through the capillary bed, which is going to drain into the venules here. Okay, you can see the branching in this little microcirculation network. True capillaries are drained at the distal end of the capillary bed portion of the central canal called a thoroughfare channel. The thoroughfare channel lacks precapillary sphincters and reflects the fact that this is the drainage end of the capillary bed. When the precapillary sphincters close, blood flows straight from the metarteriole to the thoroughfare channel and into the venule without passing through the true capillaries. Blood does not flow backward into the true capillaries from the thoroughfare channels because pressure gradients favor movement of blood in one direction only. So this would be bypassing the entire capillary bed, and you're just going through. This is if the capillary does not need to be perfused. And here you can see these precapillary sphincters coming off the metarteriole here at either end. So you're just bypassing. These sphincters are closed. Tissue perfusion is going to be regulated by local factors within each individual tissue. This is a phenomenon known as autoregulation or self-regulation. This ensures that the correct amount of blood is delivered to match the tissue's level of activity. Two main types of local autoregulatory controls exist, and that is myogenic mechanisms and metabolic controls. With the myogenic mechanism, this counters changes in blood flow by altering the arterial resistance. The velocity of blood flow is related inversely to the resistance. So if resistance increases, velocity decreases, and vice versa. The mechanism slows the blood flow by increasing resistance when the arterial pressure rises. Mechanism speeds up the flow by decreasing resistance when the arterial pressure lowers. Both changes will maintain local tissue perfusion at a constant level. This ensures that gases and other substances continue to be exchanged in adequate amounts even with fluctuating blood pressure. On the other hand, with metabolic controls, this is mediated by chemicals that are present in the interstitial fluid surrounding the capillaries. Chemicals are the result of cellular, metabolic, or ATP generating activities. In cells producing ATP rapidly, low concentrations of oxygen and high concentrations of carbon dioxide and hydrogen ions cause smooth muscle cells of the local arterioles to relax, dilating the arterioles, increasing perfusion, and ensures adequate oxygen and nutrient delivery to the metabolizing cells. In cells that are producing ATP slowly, high concentrations of oxygen and low concentrations of carbon dioxide and hydrogen ions will cause constriction of arterioles and a decrease in tissue perfusion. Each organ has its own unique requirements for blood flow and necessary nutrients. And let's look at these. The heart receives about 5% of the cardiac output via coronary circulation. The perfusion pattern is opposite the rest of the systemic circuit. Heart tissue perfusion decreases during systole. Main local autoregulatory mechanisms of the cardiac muscle tissue appears to be the metabolic controls, primarily concentration of oxygen in the cardiac interstitial fluid. Low interstitial fluid oxygen levels will trigger the production of chemicals called vasodilators, which will directly dilate the arterioles serving the myocardium, greatly increasing tissue perfusion. The brain, on the other hand, is extremely intolerant of ischemia. Sudden decrease in tissue perfusion to the brain will result in loss of consciousness within seconds. The brain only accounts for about 2% of the total body mass, but it actually receives about 15% of the total cardiac output. Autoregulatory mechanisms include myogenic and metabolic controls, maintaining cerebral blood flow at nearly a constant rate of about 750 mils per minute. Despite the constant blood supply, blood flow within the brain to different areas varies considerably with neuronal activity. This is in part due to standard metabolic controls. Areas of the brain that have higher activity consume more oxygen and produce more carbon dioxide. Certain neurotransmitters are direct vasodilators themselves. Others act on the astrocytes, which are part of the neuroglial cells, in contact with the cerebral arterioles. When stimulated, the astrocytes will induce the production of vasodilators, opening up the arterioles, increasing the perfusion to the active regions of the brain. Skeletal muscle, this is where blood flow changes dramatically, and you can actually see tissue perfusion significantly increase with exercise. This increase can be as much as 50-fold, known as hyperemia, mechanism that lies within the structure of the arteries supplying the skeletal muscle. Your feed artery, which is an artery that enters the skeletal muscle, will branch into multiple arterioles. They will also end in terminal arterioles that supply the capillary bed. During resting conditions, resistance in feed arteries is high, and many terminal arterioles are constricted. This limits tissue perfusion. When exercise begins, metabolic conditions around skeletal muscle fiber will dilate the terminal arterioles. This will produce small increase in tissue perfusion as more capillary beds open, but not enough increase to meet the demands of vigorously working skeletal muscles. As exercise continues, vessels proximal to the terminal arterioles, which are referred to as the feed arteries, also dilate, dramatically increasing tissue perfusion in the skeletal muscle. Progressive stepwise vasodilation allows the skeletal muscles to precisely match the perfusion to the degree of exercise. Another area that we look at is the skin. This is the largest organ in the body. Blood supply is located in the dermis. Oxygen and nutrients must diffuse from the capillaries in the dermis to the epidermis. Local autoregulation of the skin's blood flow takes place in response to temperature. For example, placing something warm directly on the skin will cause for vasodilation. Cold will cause for vasoconstriction. Most important control over the skin's blood flow is via sympathetic nervous system. Neurons regulate blood flow to the skin as part of temperature regulation. The sympathetic nervous system will alter skin perfusion via vasoconstriction to conserve heat or vasodilation to release heat. So looking at capillary pressure and water movements. Movement of water across the capillaries driven by filtration. Movement of fluid by force, such as pressure or gravity. The pressure at work at the capillaries. There are two basic pressures that drive water movement within the capillary. That is hydrostatic pressure and osmotic pressure. They promote the movement in opposite directions. Hydrostatic pressure, which is also referred to as blood pressure, drives water out of the capillary. whereas osmotic pressure generally draws fluid into the capillary. The osmotic pressure is due to all the solids and proteins that are found within the capillaries or in blood. Hydrostatic pressure, this is the force that exerts on the wall of the container, such as the blood vessel. Blood is fluid in the vessel that is creating hydrostatic pressure. Therefore, blood pressure is equal to hydrostatic pressure. The fluid flows from an area of higher hydrostatic pressure to an area of lower hydrostatic pressure through capillaries until the gradient is extinguished. Passive process known as filtration. The hydrostatic pressure in capillaries will change from its arterial end at about 35 to its venial end at about 15. The hydrostatic pressure of the interstitial fluid is so low that it's functionally typically somewhere around 0. Thus, there are steep hydrostatic pressure gradients on both ends of the capillary that drive water out of the capillary and into the interstitial fluid. So here, if you look at the arterial end, you have hydrostatic pressure of 35. Remember, in the interstitial fluid, 0. Even at the venous end, you have it at 15. So the normal movement is going to be water out on both ends. But the problem is we have this osmotic pressure. This is the force we must apply to a solution to prevent water from moving into it by osmosis. Solution with higher osmolarity also has a higher osmotic pressure. Osmotic pressure of the capillary blood is about 25. This is created almost exclusively by the large proteins that are present in the blood, especially the protein albumin. Proteins are too large to leave the capillaries, so the osmotic pressure remains consistent throughout the capillary's length, and the osmotic pressure is very low in interstitial fluid. This difference in osmotic pressure creates an osmotic pressure gradient known as the colloid osmotic pressure, or oncotic pressure. The colloid osmotic pressure draws water into the capillary by osmosis. So now if we look at this, we have an osmotic pressure of blood of 25. So this is going to draw water into if the interstitial fluid is at 0, okay, at both ends. Now let's put the hydrostatic pressure and the osmotic pressure both into the scenario here. At the arterial end, the hydrostatic pressure is 35. The osmotic pressure is 25. Remember, these are opposing forces. So the net filtration pressure, which is the difference between the two, is going to be 10. And that's going to force water out. At the venous end, the hydrostatic pressure is 15. The osmotic pressure is 25. The net filtration pressure is negative 10, which means you're going to have the osmotic pressure drawing water in and fluids will move back in. So what you have is fluids moving out at the arterial end and fluids moving in at the venial end. And that is what we refer to as the capillary net filtration pressure. And that's sort of defined here. Okay. So a simple visual analogy for hydrostatic and osmotic pressures. Imagine the hydrostatic pressure as a faucet within the capillary that is always turned on so that water is always flowing out. Next, imagine the osmotic pressure as a sponge within the capillary, constantly drawing water into it. At the arterial end, the faucet is turned on more strongly and spouting out more water than the sponge can reabsorb, so the capillary loses more water. At the venial end, the faucet is turned down, putting out less water so the sponge is able to reabsorb more. So at the venial end, the capillary gains more water. Over the entire length of the capillary, the faucet is going to spout out more water than the sponge can reabsorb, so there's a net loss of water from the capillary. Okay, and that's what's shown here. Okay, so if we look, the hydrostatic pressure, the colloid osmotic pressure is negative 22, the hydrostatic pressure is 35. Your net filtration pressure is 13, you're moving out. The hydrostatic pressure at the venule end is 15, osmotic pressure, colloid osmotic pressure is 22, that's -7, so the pressure drawing fluids back in is lower than the pressure drawing fluids out. And what that means is you're going to end up with more fluids out here. Now, those fluids have to be returned. They have to be returned, otherwise eventually your blood will run out of fluid. And that's where your lymphatics, which is going to be our next chapter, is going to come into play. They will pick up that residual fluid that remains in the interstitial fluid that was not returned back in at the venous end. Now, edema is characterized by excessive amount of water in the interstitial fluid. Common cause of edema includes an increase in capillary hydrostatic pressure gradient due to hypertension or decrease in the colloid osmotic pressure due to liver disease, cancer, or starvation, among other disorders. Peripheral edema, this is pronounced edema, is found in the hands and feet where hydrostatic pressure gradient is already slightly higher due to the effects of gravity. Even more profound edema may result from a decrease in colloid osmotic pressure where excess water tends to accumulate in interstitial fluid of the abdominal area, which is known as ascites. Okay. And that is the end of recording 3.